Thank you again, Dr. Chang, for that great presentation. So my name is Gaurav Bhattacharjee. I'm a regional marketing manager for the West here at 10x Genomics, and it is my pleasure to introduce Dr. Pham. And Son, if you want to come on up here. So Dr. Pham is the CEO of BioTuring, and we're particularly grateful that he was able to sub in for us on relatively short notice, as we did have one of our speakers had to cancel due to health reasons. So thank you again very much, Dr. Pham. He'll be speaking today on a simple platform that makes single cell and spatial data analysis truly accessible.
Good. You hear me? Is everything good? Good. Let me check. Can I have the... So it can be a very good talk, but not likely. It can be the worst talk of today. Very short and full of embarrassment. It can be just 5 minutes talk. And then I feel myself embarrassed for the next several weeks. Let me explain you why. So we are the computational people, and we don't have so much slides to show. So instead of showing code, it is so complicated. In this journey, let's try to explore a series of important biological questions. And let's try to see how I deal with that using BioTuring software. If it is good, then we can spend 30 minutes from 10x Genomics. If it's not, then I lose this game, and the next, I stop my presentation in 5 or 7 minutes. But just in case, that we have some slides. So about my journey started with mathematics, and then I went to explore biology with Pavel at UC San Diego. And then at some point, we trying to taught the first bioinformatics course on Coursera with about 20,000 students enrolled. Was the first one there. And then I went to Salk Institute for doing a postdoc in the wet lab. And about 1.5 years, I finished my postdoc, and I went for, instead of faculty, I started the BioTuring about 6 years ago. And right now I spend most of the world of pharmaceutical companies already. And we start rolling out to academics. So this is the first time probably that I introduced this platform to the audience. So let's start. I have to say something, because just in case that we need to spend some time, just in case the games failed, right? So seems to be that I cannot click. Yeah, you see, the game is not that easy. Okay, I start to see some. Need to go over here? Okay. Okay, he got me, right? You see, it started very difficult. So, Serge, about several years ago, he said that this is the century, the 21st century is the century for biology. Why is that? Well, you see, with this, before single cell, I was a bioinformatician's life very easy. We have several dozens of samples. Maximum, we have hundreds of samples. But 10x genomics, we have to blame that they brought the game more difficult. Now we start seeing the labs start generating hundreds of thousands of cells or millions of cells that we are talking about. So you see this log scale here is 10x log scale there was started by 10x genomics. And the game is not that easy anymore. Now we have to deal with millions of cells. And just right after that, you can see the huge project from Human Cell Atlas, from 500 labs. And now they have generated and curated about 36 million of cells. So these scales in generating data are actually making the game for bioinformaticians more difficult. It creates a lot of computational bottlenecks. Well, I would agree with Serge that this century can be the biology century. But about a decade ago, when we work in computer science, we told that this century is the century for accelerated computing. And thanks to that, let me show you a very similar graph that we plot. That is the floating point instruction per second. The blue lines underlined is the line for number of floating point operations, additions, multiplication of floating points that we can perform per second. And about 2010, we start seeing we cannot increase anymore the speed of CPU. Luckily, we see the green light that started as another game, another player in computer science. It's called NVIDIA. That generated suddenly, the number of floating point operations that you can do per second scale up thousands of times. This is the start of AI. This is the place where artificial intelligence was born. So I think this century can be the century of biology, and in computer science, this is the century for accelerated and AI computing. Taking that advantage, BioTuring has been doing a very silly job. What is it trying to do? Trying to invent the wheel. Taking the algorithms that was written and optimized for decades. You can see PCAs here, and you can see new algorithms, t-SNE, UMAP, Low-End clustering, KNN, K-means, correlation, harmony, AU enrichment, and many more. We start computer science data structure 101. But this is for the new century. How to scale these algorithms when we have tens of thousands of parallel computing units rather than single or several CPU cores. As a result, we obtain thousands of times faster. Now, we can analyze single cells, millions of cells. Instead of waiting for a week, like you may now experience, now it can be less than a minute in real time. So this is the benchmark, one particular thing. For a data set of size 1.7 million of cells, we just need about 56 seconds to run the full pipeline from start to end. We compare with other packages, and it scales thousands of times faster. But how about comparing with the one who actually invented the hardware, which is NVIDIA, and they have the software part as well. So NVIDIA Rabbits, which is the software written by NVIDIA, and if you can look at those graphs, On the left side is the time. It takes NVIDIA rabbits about 2,000 seconds, while it takes by Turing algorithms about 13 seconds. For the memory usage, instead of taking 40 gigabyte, for Turing just 3 gigabyte. With this, instead of having 36 million cells, very quickly, because we are now having ability to analyze single cells in real time, we quickly collect, rerun, and curate all the currently publicly available single cell data. And the current database is 118 millions of cells right now. And one thing that it takes a little bit more time to do is standardize metadata. So of course, people use different words to describe the same thing. So all the cell type, condition, development stage, and other things are standards into standard vocabulary. Why we should do that? In order to pull things together. With this huge 1,000 times accelerated computing, now, and standing on hundreds of million cells, now we are able to redefine cell types from the data, not from 5 million size of data, from hundreds of millions data, all currently available single cell data that we can cache. And this has two things. One, theoretical framework that Connor and also Aviv, who actually define the theoretical framework for redefining cell types, signatures. Essentially, this problem is also an NP-complete problem. So essentially, to solve this problem, one has to enumerate a huge possibility, which is almost impossible when you have the normal CPU world. I don't know whether it's possible when we can solve it in the quantum computing yet, but for now we still don't know. But when we accelerate thousands or tens of thousands of times the algorithms, we have the ability to enumerate much more many combinations. And one of the colleagues here actually working in the very efficient suffix array data structure to enumerate trillions of combinations in order to define cell type signatures. And another colleagues actually from the data and from the algorithms actually give the first cell type prediction algorithms not on several millions of cells, but was trained on hundreds of millions of cells. I do not know any algorithms that, other than this, that can scale for the size of hundreds of millions of cells yet. And by the way, the name here was her son's name and was born during the course of she's working on the algorithms. So we spent almost, 7 minutes or 8 minutes talking about the preliminary. And now let's go to the real part. And this can be very embarrassing for me because we start asking real questions, or you can just start asking me questions and I start answering it. But to make it really simple, let's try to go to the system. Let's try to explore. So this is a very simple interface here. And you can start coming here and just start query a gene, like CD3D, for example. So here you can see, this is the cell ontology, right? Where the redness actually expressed where the gene expressed in the cell or not. So you can see CD3D expressed a lot in the subpopulation of T cells. And let's try another one, FOXP3. It expressed a lot in regulatory T cells. And let's try another one, another gene, RBFOX1. It expressed a lot in neuron cells, right? So let's start with, and this is, you search for a gene. Now, you're talking to a system of hundreds of millions of cells beneath from multiple technologies here. Now, let's try to search. How about searching for, let's find all the cells that express a gene CD3D. Well, how would we do that? Well, we would go filter, expression profile, and enter CD3D. And then press search. And you can see it returns how many? It returns 873 studies that have cells that express gene CD3D. But this is probably too easy. Let's try to make it a little bit harder. Let's try to add another gene. So we want to find cells that express two genes, CD3D and CD8, for example, okay? And you can search. So this is CD8 T cell. And you can start seeing some points that are not red here, gray point here. The cells, each cell is a point here, and the red one is the one that mush, and the gray one is the one that's not. This is easy. Let's make it a little bit harder. How about excluding a gene? The cell express CD3D and CD8A and not express some genes that you may like. Let's say CD4. And click on it, it turns red. What are we searching? We are searching for cells in hundreds of million cells that express CD3D, CD8A, and not express CD4. So this is the result. It turns you how many? 677 studies. But this is search, right? But the search actually not stopping at searching for genes only. It can search, you can see Google Lens, it can search for pictures. Here, you can search for cell populations. Imagine when you have a data set, you see a strange population of cells like this, you ask yourself, oh, has anyone found these populations or not? Let me try to do it, for example. Let's choose this exhausted T cell and trying to search what it's doing. It takes the expression of this population and it asks a question. Has anyone found any populations of cells that are similar to this current chosen populations? Well, this is the result. It says, it captured this is CD8 positive alpha beta exhausted T cell, which makes sense. It found 5 studies. Let's explore it a little bit. So this is the population. The blue one is the population that we are searching for. We start with the search. And these 5 populations are the populations that it found from, it has GSE ID here. If you need, you can just click and you can jump to that study to further explore it. Okay. But let's see, it found 5 populations. And let me do something. Let me combine them into one single study and see what it found. Well, it mostly, from on these 6 studies, mostly are exhausted T cell, which makes sense. We know originally that this is exhausted T cell. Of course, the algorithm didn't know, but this is exhausted T cell. So it means that the cell search actually bypassed the technical effects and moved to the similarity in cell type. Now, if we move to conditions, this is neoplasm of epithelial tissues. And the population that we searched for was the exhausted T cell in cancers, right? So it not only captured the cell type, but it also captured the conditions where the cell type come from. Okay. Now, let's make the question a little bit more difficult. Let's try to search for the same thing, but let's try to explore the breast cancer, any breast cancer data set here. So the good thing is that we have all the metadata standardized. So now you can go and search for tissue, and you search for breast cancer. And you can search for it. So you can combine the search with genes and metadata. Now you can search and you found this is a cell, a single study. Imagine that you're having this single study. In this study, it is actually a single cell map of intratumoral changes during anti-PD-1 treatment of patients with breast cancer. And, we have all functionality from pathway, pseudo-time, clonal time, marker genes, differential expression compositions here. But and we can read the papers together with, let's check whether the authors are actually saying right in the paper, right? But now with this, we can check. So let's focus on T cell. Let's focus on T cell now. And let's ask a question. Let's ask a question. We have T cell here. We have, let's say, sampling time point, and say respond to treatment or not. Okay. Let's generate some charts so that we understand what it is. So we have, this is on treatment, and clonal expansion versus non-response. Let's search for a gene that the authors report that in T cells, the CD1 actually express more than in T cell of the patient that respond to treatment. And let's search for it. And you can see, this makes sense, right? The non-response, the gene doesn't express so much. So this actually, the author in this paper actually focused a lot on immune cells. But if we take a look here, there are a lot more cell type than just immune. Let's switch to the author cell type so that we can see. Yes, the fibroblasts can be very interesting. In this study, there's about 175,000 of cells. And let's focus on the fibroblasts example. And let's perform subclustering from them. Now we have fibroblasts. You can run t-SNE UMAP. Rather than waiting for half an hour, for the pipeline to run again, now you can ask a question, change parameter, and press run. And in several seconds, it will give you the result again. Right? So this allows very interactive, ask question, answer the question immediately. But it requires a lot of hard work in computer science to be able to scale thousands of times for the algorithm to work on that. So this is fibroblasts. And now we can ask the question, a more interesting question, whether there's any difference in the fibroblasts of the patient that responds to treatment versus non-respond to treatment. And let's say time and respond to treatment. And we can say, okay, what are the difference in the fibroblast? This is clonal expansion. This is, I say, response. And this the second one is the non-response group. Non-response. And I say run. What is the difference of fibroblast in non-response versus response after the treatment? Let's sort by log-fold change. And you can see quite a lot of marker genes here that CXCL11 chemokines genes here attracting T cells coming to the fibroblast in the case that it responds to treatment. And then it raises another questions. Well, this is one data set, right? Can you combine all the fibroblasts in cancer and tell me what is the pattern? Not in one study. In one study, it may be, generating from having a, when some scientist forgot something, it may not be significant. How about, is there any pattern that consistently appears from data set to data set? Well, in order to have that ability, you need to have the ability of finding all fibroblasts in cancer. How would we do that? Let's try to do it again. Study. Let's go to filter. What do we want? We want cell type, fibroblast. We want the condition to be neoplasm of epithelial tissue. What we want the sampling site to be adjacent normal and primary tumor. Now we ask question. What are the differences between fibroblasts at the primary tumor versus the adjacent normal site? You can search. It returns 55 studies. Let's combine all these guys together into one silico study and to see what is happening. Create a custom data set, fibroblast in cancer, and create. Okay, and done. And this is fibroblast. Let's switch to signature genes. That's trying to find the signature genes between sampling site, between the primary tumor versus adjacent normal tissue. And let's change the parameter a little bit. Let's find signature genes. So now we are talking about not one single data set. We are talking about 55 data sets right now. And if you look at it, you can see some genes, very interesting gene here, postin. You can sense the FAP, fibroblast-associated proteins here, and which is reported in a lot of places. And there are a lot of other genes that are not reported. And the questions there is, now this is not from a single study. This is a combination of multiple studies. Now, let's try to do one more thing. This is the fibroblast activated proteins, right? In the where it is at the primary tumor. Let's try to search for that gene, FAP. FAP. You search for a gene, you know that it expresses quite a few in fibroblasts. But let's click on 10x Visum. Now it is on the slides from 10x Visum's data here. You can see on the left, actually two parallel panels. On the left is the histology image. On the right is the histology image superimposed by the expression grid. Let's go to one more. This is in the press, right? And if you, let's turn the opacity down, trying to explore this a little bit. So this is the cancer part. This is probably another cancer part here, right? And this, and let's search for the gene Pham, F-A-P. And let's increase the opacity a little bit more so that we can see, okay, so there are quite, the unit is quite, so there are quite a few here. So this drives me to ask questions about the MyCAF and ICAF. They're actually two important cancer-associated fibroblasts. And the marker genes for MyCAF is ACTA2, I think. And let's see. This is very important. So you can see this region of cancer doesn't express the MyCAF signatures, while at the same time, in this region, MyCAF actually expressed a lot, which is representing this additional cancer-associated fibroblasts. And very interesting when you search for CD3D, you can see here a lot of T cells cannot infiltrate. And on the other side, in the absence of MyCAF, T cells can infiltrate a lot in the place. And then you go back to the platform and say, oh, this is Visium technology. And here's Xenium. Where's Xenium? Well, Xenium is here. You can explore. This is another study in breast cancer as well. This is Xenium. And you can see the Xenium. The same patterns actually appear, but this round, you have the single subcellular expression of that. So, of course, you have cell-cell interactions and other functions, but the same patterns can be repeated in using the Xenium data. And, in case there's actually an additional tool that you can use, for example, you can search for D or CD3E. And let's try, you want to plot something. Let's try to see how do you plot something here. Let's say cell type. And BioVinci is the platform that supports scientific visualizations. So essentially what it means is that you can just drag and drop a column and create different box plot, different ways of visualizations here. You can edit just by dragging the visualizations, and when you want to change, instead of remembering coding, now you can change the parameters here. So this is an additional tools for helping biologists explore the data. So I was lucky that I could went through the time of this game, able to finish it, without problems. But I think going back, 10x Genomics are generating a lot of data. It's the first one who boosts the number of data points 2,000 times. Luckily, in computer science, NVIDIA and other companies had prepared for that about more than 10 years ago. Having new, instead of the normal computing paradigm, it born a new GPU computing, 1,000 times speed up, and therefore we are now able to see the advancement of AI. It's from a high performance computing there. And I think this is a century of accelerated biology, also the century of accelerated computing, and allows scientists to have the power to be able to analyze their data in combinations of all existing data in the world in real time. Thanks so much. Questions?
Hi, very great talk. So I have a question because I think one of the common problem that people will encounter, especially when compared with public data set, is that there's batch effect. Also, there's like different kind of sequencing depths. So just wonder, how do you cope with that problem so that you have a reliable readout across the board?
Yes, very good questions. So as you can see, the most reliable, I think, is also speed is also good. It runs about an hour to finish for batch effect was the harmony, which is written by Dmitry at the Broad Institute. And at BioTuring, we actually scale that to thousands of times. So instead of waiting for an hour, you wait for seconds for combining data set together. But this is even a deeper questions. So harmony was designed to combine like several data set together. It was not designed to combine thousands of data set together. Of course, batch effect is always a problem there. And that's why when we started working on this about 4 years, and we tested a lot of algorithms, but one day, very simple ideas, I think it helps a lot, is change the unit. Instead of using UMI, instead of using whatever unit it is, convert them into a ranking system. So the idea here is that regardless of the technology, if gene A expressed larger than gene B in the sample, the rank should be preserved in that. So it is a more stable. I'm not saying that it's not without problems. Of course, ranking can change, but it is present much more stable way to when you combine data set together.
So
yes,
thank you for the presentation. Then I think in the R, the serial R in Python program, there's algorithm called the integration and label transferring.
Yes.
To annotate the cell population.
Good.
So will the BioTuring do that job to annotate the new data set, your own new data set?
Very good questions. Very good question. So Seurat and other package they transform the data. And essentially at BioTuring, we take actually a different approach for cell type labeling, which we need a lot of time to talk about that. But essentially, the heart of that actually comes from the theoretical framework that Aviv actually present in the paper in 2019 about the connected with the gene panels part. But essentially, that one is an NP-complete problem. That provide very accurate solutions. But unfortunately, an NP-complete problem is a problem that is extremely difficult to solve. It may take hundreds of years to lead to an optimal solution. But also thanks to the new paradigm of computing, where instead of having one core, we have now hundreds of thousands of cores. And the challenge is how to design the data structure that allows to search through that space. But we need to discuss a lot more on that.
The second question is, in the more publication coming up, in a lot of disease tissues, people discover the unconventional T cell at its transition state. Can your model, deal with that?
Very good question. And it lies into, if you can explore with that, it lies into the cell search functions of the ability to take a population. If you look at the Google Lens, you take a picture, you doesn't tell this is a bag, but this is unconventional bags, right? So and you take this picture, you search for all the image, it returns you bags, similar bags. Of course, it can be another 10x bags that is there, but when we designed the cell search, we did not aim to rely on the author's curation, but this is unbiased where we don't take the annotations into that. So when you search for an object, you don't need to tell what this object is.
So I think that relates to one of the two questions we have online from our virtual platform, and that was around identifying rare cell populations. So I think
Yes.
the same thing applies. Which package would you use for that? And then another related question was, can you use it, or can you use TalkTo data to analyze multi-omics data?
Good, good. So the first question is already answered. The second questions of talk-to data used to analyze multi-omics data. You can see we have single cell, we start have the ataxics, we have clonotype data. We already have Visium, and soon we will see a new wave of Xenium data. This is a huge wave of single cell spatial that we are collecting into the talk-to data.
Thank you.
And also what happened when we have so much data like that? Probably quite a few of you explore ChatGPT of typing something and give you answer. Of course, a lot of time it gives you incorrect informations. So what we think in the future is that we're collecting a lot of data, build extremely fast, high performance computing. And on the top of that is the intelligent part. And then later on, you come and you ask a research question instead of relying on the language model only. It touches the real data, pulling from it, and give you some directions to explore. And this is where we are heading to. Thanks so much.
So thank you very much again, Son, for that excellent presentation. And now we are having our panel come up. I'll let John